Welcome to the deep dive. Today, we're looking at a really fundamental question in marine science. How do we actually count fish reliably? I mean, for years, the main tools, things like trawling nets, acoustic surveys, well, they're costly.
Definitely costly
Uh,
and resource intensive, invasive too. Plus, you just can't use them everywhere. Think about rocky bottoms or really sensitive habitats.
exactly. So we're diving into a paper today that offers a pretty clever way around some of this. It's not about a new net, it's about using genetics.
Right.
Paper's called Predicting Trawl Catches Using Environmental DNA. It's in the ICES Journal of Marine Science, lead author Guri and the team. And it feels like a big step towards making marine monitoring, well, cheaper and maybe smarter.
Yeah. And the context here is super important for anyone listening. If you're talking fisheries management, conservation planning, you need good numbers. Biomass, diversity, where things are.
Yeah.
It all depends on decent population estimates. And environmental DNA, or eDNA, that's the genetic stuff organisms leave behind in the water. It always had this huge promise because it's not invasive, right?
Yeah.
You just grab a water sample.
Simple in theory.
Simple in theory, yeah. But the massive roadblock for ages was this. The standard technique, metabarcoding, it could tell you what species were in the sample, relatively speaking.
Right.
Like, 10% of the DNA reads are cod, 5% are herring.
Okay.
But it couldn't tell you if that 10% meant 10 cod or 10,000 cod. It gave you relative proportions, not absolute numbers. And that really held it back from being integrated into, say, fisheries stock assessments. It was stuck being qualitative.
Okay, right. So the mission here for Guri and the team was basically to crack that code,
Yeah.
to build a kind of molecular translator, yeah. Something that could take those relative DNA reads and turn them into solid, absolute estimates of fish density,
Precise.
moving eDNA from just, is it here,
Yep.
to how much is here.
Yeah.
A truly quantitative tool.
Exactly. And they knew you couldn't just compare trawl numbers directly to water sample DNA counts. There are different units, different processes involved.
Right, apples and oranges.
Totally. So they needed a sophisticated statistical bridge. That's this comprehensive Bayesian joint model framework they developed. Its job was basically to force the molecular data to calibrate itself against the physical counts, the known quantities.
And this wasn't just a benchtop experiment, was it? They took this out into the real world.
Oh, yeah, absolutely. They based the study in Balsfjord up in northern Norway, pretty long fjord, high latitude. And they collected data over 3 years, October 2019, 2020, and 2021.
3 years, that's significant.
It is. And the complexity came from pulling together data from 5 distinct sources. That's what the joint model had to juggle simultaneously.
Okay, let's break down those inputs because this calibration part, that feels like the real innovation here. So obviously the traditional trawl counts, the physical baseline.
Yep, that was the reference point.
Yeah.
Using a standardized camp on trawl. But then for the molecular side, they needed checks and balances. So first, they used droplet digital PCR, ddPCR. Think of it as a very precise way to count DNA molecules for a specific species.
Like a molecular measuring cup.
That's a good way to put it. They did this for Atlantic cod and herring in the environmental samples. That gives you an absolute DNA concentration for those key species.
Okay, so that anchors the molecular data for cod and herring. But what about all the other species the metabarcoding picks up? How did they make sure those relative reads were accurate across the board?
That's where the internal yardstick comes in. They used what's called a mock community. Basically, a pre-made cocktail of DNA from 10 different fish species mixed together at known concentrations. They only ended up using 6 in the final analysis, but the principle holds.
Why do that?
Because when you sequence a mix of DNA, some species' DNA just amplifies better. It's louder in the data. It can skew the proportions.
Like some voices carry more in a crowd.
Exactly. So by sequencing this known mock mix, they could measure those species-specific amplification biases. They learned how much to sort of digitally turn down the volume on the loud ones and boost the quiet ones to get the true proportions back.
Okay, now I see how the joint model fits together. It takes the raw metabarcoding reads from the fjord water, corrects them using the bias info from the mock community,
Right.
and then scales everything up using the absolute numbers from the ddPCR for cod and herring as reference points.
Precisely.
Yeah.
That whole process allows the model to translate those initially relative DNA reads into estimated absolute concentrations for all the species detected.
And that calibrated eDNA concentration is what they then link to the trawl data.
Exactly. That calibration is the foundation. And the model, it successfully gave them empirical estimates for the key link between that eDNA concentration and the actual fish abundance seen in the trawls.
Which brings us neatly to the star of the show. This conversion parameter, theta.
Yes, the theta. It's kind of the magic number, or maybe the fudge factor, but in a good calculated way. It encapsulates all the messy, real-world stuff that happens between a fish swimming around shedding DNA and a net catching some fish.
So things like how easily the trawl actually catches a certain species.
Yep, trawl catchability.
How much DNA that species sheds.
Shedding rates, degradation rates in the water, dilution, transport, even how efficiently they recovered the DNA in the lab. Theta bundles all that complexity into one value.
Defined as, what were the units again?
Fish per square kilometer per DNA, copies per microliter. So fish density from the trawl linked to eDNA concentration.
So this is the big leap then. It's not just saying more DNA means more fish nearby. It's providing a statistical framework to actually predict the trawl catch density using only the eDNA data, at least for out-of-sample tests.
That's the breakthrough, moving from correlation to prediction, and crucially because they corrected for the molecular amplification bias first.
Using the mock community.
Right. The resulting theta values, which varied between species, actually start to reflect real biological differences. Differences in shedding, or maybe catchability between species.
Okay, let's talk results. How well did this prediction actually work?
Pretty well for most species tested. 5 out of the 7 main species showed a strong correlation between the predicted densities from the eDNA model and the actual observed trawl densities in the test data. The model fit was good, indicated by low delta values.
And you mentioned the theta values varied. Can you give an example?
Sure. For Atlantic herring, the log of theta was around 595. But for a smaller species like capelin, it was much higher. Log theta of 12.96.
Wow, that's a big difference. What does that suggest?
Well, it could mean several things. Maybe smaller fish like capelin shed proportionally less DNA for their numbers, or their DNA degrades faster. Or perhaps they are harder to catch efficiently in the trawl relative to their true abundance compared to herring. So the model needs a much larger conversion factor to match the eDNA signal to the trawl catch for capelin.
It really drives home that this isn't a one-size-fits-all conversion. You need species-specific biological context.
Absolutely. It reinforces the need for biological knowledge. It doesn't replace it. But, we really need to talk about the 2 species where the model didn't work well. Those failures are incredibly informative.
Agreed. Let's tackle those. First up was lumpfish.
Yeah, lumpfish, Cyclopterus lumpus. The issue there was the trawl data itself. They caught hardly any lumpfish in the bottom trawl surveys.
Which isn't surprising, right? They're not always caught well by that gear, especially deeper down.
Exactly. They often live below 60 meters where this trawl isn't efficient for them. So because there was almost no trawl data to calibrate against, the model just couldn't get a reliable estimate for theta dollar. The uncertainty was huge.
Okay, so lesson 1. If your traditional gold standard method misses the species, using it to calibrate a new method won't magically make that species quantifiable. Makes sense.
Right. Garbage in, garbage out, essentially, for that calibration step.
But the other failure, European place, was kind of the opposite problem, wasn't it?
It was, and really striking. For place, Pleurontex platessa, the trawls did catch them. They observed relatively high densities, actually. It's a flatfish, lives on the bottom. You'd expect the trawl to get some.
Okay.
But the eDNA metabarcoding data came back with 0 reads, nothing, no genetic signal for place at all, despite the fish being physically caught nearby.
Whoa, so what does the model do then?
Well, it tries to calculate theta, but because the eDNA concentration is 0, the math basically breaks down. The lambda term related to DNA becomes unmeasurable, and the resulting theta estimate is astronomically high and meaningless. Log theta was estimated at over 30.
So 2 key failure modes. One where the traditional gear fails, one where the eDNA seemingly fails. Does that mean this whole approach only works for species that are both easily caught by trawls and shed lots of detectable DNA? Is it a limited fix?
I don't think so. I think what it does brilliantly is expose the specific weaknesses in both monitoring approaches species by species. The lumpfish case highlights trawl limitations. The place case flags a major issue with eDNA for that specific species in that context.
Could be very low shedding rates, or DNA degrading instantly on the seabed.
Could be. Or maybe primer issues, though less likely given the controls. The point is, the framework itself worked. It just showed that for place, you need both types of data present to make the link. It tells researchers exactly where they need to dig deeper. Why isn't place eDNA being detected?
Okay, that makes sense. It pinpoints the problems rather than invalidating the method overall.
Right. And if we zoom out, again, thinking about the bigger picture for management, This framework offers real advantages. Traditional surveys face bottlenecks, vessel time is expensive, weather dependent, some areas are inaccessible.
Marine protected areas, for example.
Exactly. eDNA sampling is potentially much cheaper, faster, less impactful ecologically. You could sample more often in more places. This model provides a way to make that cheaper data quantitatively useful.
And the authors point towards future refinements too, right? Making it even more precise.
Yeah, they suggest moving beyond these broad fjord-level averages. The framework could potentially allow for site-specific or even depth-specific estimates if the sampling resolution is there. That would be a huge gain in accuracy.
But they also flag limitations that need more work.
Definitely. A key one is that the current theta value bundles together the trawl's catching efficiency, what they call FEV dollars, and all the DNA-related factors, which they group into lambda. That's shedding, degradation, dilution, etc.
So right now, you can't tell if a high theta is because the fish is hard to catch or because it sheds very little DNA, or both.
Exactly. So a crucial next step is to try and estimate the trawl catchability independently, maybe using other methods or historical data.
Yeah.
If you can pin down dollars, then you can isolate lambda.
And understanding lambda gets you closer to the pure biology of the eDNA signal itself.
Precisely. How much DNA is actually being produced and how long does it stick around? That's fundamental ecological information.
And what about fish size? That seems critical for fisheries.
Absolutely critical. They highlight that future work needs to tackle allometric scaling, basically. How does the size of a fish affect how much DNA it sheds, if you can crack that?
But then eDNA might tell you not just how many fish, but roughly how big they are.
Potentially, yes. Getting population size structure from water samples would be a massive leap forward for stock assessments. That's a really exciting direction.
And they also mentioned the model's flexibility. It's not just tied to trawls.
Right, the statistical architecture is adaptable. You could, in theory, replace the trawl data component with data from acoustic surveys, or maybe even catch and release studies. It provides a template for integrating eDNA with various monitoring techniques.
So wrapping up the core achievement here.
I think it's that this study provides the first really solid empirical demonstration of calculating these species-specific conversion factors between eDNA metabarcoding data and traditional trawl data. It takes eDNA from being this cool but mostly qualitative presence-absence tool
Into something genuinely quantitative that can actually plug into the models used for managing fisheries and ecosystems.
That's the leap.
So what this means for you listening is that monitoring our oceans, which is vital for conservation and sustainable use, could become significantly cheaper, less invasive, and potentially more comprehensive. We might get better data more often, which helps everyone make smarter decisions.
And here's something to maybe mull over. We saw that the conversion factor, TOL1, varied hugely between species like capelin and herring. If TOL1 is the link between physical catch and DNA signal, what does that massive variability really tell us about the underlying biology? Which factors, shedding rates, behavior, degradation, maybe even catchability itself, make certain species inherently harder or easier to see in the ocean, whether you're looking with a net or a DNA sequencer?